Welcome to the BioInsights, where science comes alive.
Today we are tackling a truly fascinating challenge. Imagine just looking at a scoop of everyday dirt or maybe a drop of water. To the naked eye, it's just dirt or water. But we know for a fact that there's an entire chaotic, invisible microbial world thriving right there. So the big question for scientists is this: how do we actually turn that invisible chaos into structured, countable data?
Literally, how do you count what you can't see? It sounds a bit like a riddle, but for microbiologists, it's an everyday physical problem that they just have to solve. We often think of microbiology as this purely theoretical science, but as we'll see from three brilliant scientists today, studying the microscopic world really requires conquering a whole series of very tangible, hands-on physical challenges in the lab.
Okay, let's dive into this. We've structured today's lab session into three main parts. First, isolating the unseen. Second, the math of serial dilution. And finally, aseptic spread plating techniques. Let's start right off with Section 1, isolating the unseen. So before we can study anything at all, we have to actually extract these microbes from their messy, natural environments and get them into a controlled setting. Entomologist and researcher Dr. Rini shows us exactly how this extraction begins. To find specific bacteria in agricultural soil, she takes about 2 grams of that soil and mixes it into 10 milliliters of a prepared sodium acetate solution, right inside a flask. She wraps the top securely with aluminum foil, to protect it from any outside contaminants, and then she places it into a very specialized machine overnight. Now, what's really interesting here is the specific device she uses. It's called an incubator-cum-shaker. Because honestly, it's not enough to just let the samples sit on a desk somewhere. This machine does 2 absolutely crucial things simultaneously. It maintains a perfectly controlled warm temperature, and it continuously shakes the flask. Giving it this constant physical agitation overnight is what allows those few invisible bacteria hidden in the soil to rapidly multiply into a dense, workable liquid sample that scientists can actually use. Moving right along to Section 2, the math of serial dilution. Well, by solving our first problem, we've actually gone and created a second one. Once incubated, the cells are incredibly dense. To put the scale into perspective, microbiologist Dr. Monica Oli looks at Saccharomyces cerevisiae. That's your common baker's yeast used for bread and brewing beer. Her ultimate goal is to find out exactly how many yeast cells exist in just 1 single milliliter of her sample. But here's the catch. If you were to just smear that 1 milliliter on a plate right now, it would just grow into a giant, unreadable blob. There are simply too many cells. You have to thin it out.
And this brings us to the elegant math scientists use to solve that density problem. It's called a dilution series. Think of it like a cascading waterfall of math. You line up a series of tubes, each filled with 9 parts of a sterile buffer, like PBS. Then you take 1/10 of the volume from your original densely packed sample and mix it into that first tube. Boom, that creates a 1:10 ratio. Then you take 1/10 from that new tube and move it to the next one, creating a 1:100 ratio. You just keep cascading down, 1:1,000, 1:10,000, and you are rapidly decreasing the cell concentration with every single step. But the physical action of how this transfer happens is completely vital. You move that 1/10 volume and then you use a vortex mixer, which is basically a rapid vibrating machine, to thoroughly blend the new tube. But listen, here is the golden rule of serial dilution. You absolutely must discard and use a fresh pipette tip for every single transfer. No exceptions. If you reuse the same tip, the high concentration of sticky cells from the previous tube will just cling to the plastic. That's going to artificially inflate the numbers in your next tube and completely ruin your data. So fresh tip every time. Which brings us to our final step, Section 3, aseptic spread plating techniques. Okay, now that we have perfectly thinned out samples, our final challenge is spreading them out safely. And before any biology happens here, we have to talk about safety, because we are literally about to play with fire and live bacteria. Microbiologist Stephanie Sweeteek outlines a really rigorous checklist. You must fasten all loose clothing sleeves, you must pull your hair back completely so you don't accidentally ignite yourself while leaning over the bench. You've got to clear your workspace of clutter and absolutely ensure the gas line is entirely off when your Bunsen burner isn't lit. We definitely don't want the room filling up with flammable gas. Once those safety checks are complete, we are back to facing that microscopic scale. Stephanie pipettes just 100 microliters of her pre-diluted bacterial liquid and drops it directly onto the center of a fresh agar plate. It's just a tiny bead of liquid, really, but it holds all our target cells. So how do we spread that tiny bead around evenly without introducing a bunch of new germs from the air? Well, we use a glass or metal tool, appropriately called a hockey stick spreader. But first, it has to be perfectly sterilized. To do this, you dip the spreader in a tub of ethanol and then quickly pass it through the Bunsen burner flame. The ethanol ignites, which burns away any stray contaminants. But this requires a really vital balance. You do not hold the stick in the fire to heat up the metal. You literally just want to catch the ethanol on fire. Afterwards, you let it cool for a few seconds, then you tap it gently on the very edge of the auger. If it sizzles, well, it's too hot and you're going to incinerate your sample on contact.
Once you're sure it's cool, the actual spreading technique is just deeply satisfying. I absolutely love this description from Stephanie. She explains that you swirl the bacteria across the plate, quote, almost as though I was trying to coat a pan with a stick of melted butter. I mean, it paints such a vivid mental picture, doesn't it? You have to be incredibly gentle but thoroughly complete, using this spinning motion to make sure those invisible cells are perfectly and evenly distributed across the entire surface of the agar.
Now, comparing these methods really builds a larger picture of how lab methodology works. We've explored two very distinct plating solutions today. On one side, we have Dr. Monica plating yeast. Because of the rapid Petrifilm she uses, she plates a full 1 milliliter and simply presses it flat using a small plastic stomp device. But on the other side, Stephanie is plating bacteria, using just 100 microliters on traditional agar, which requires that thrilling flame sterilization and careful swirling technique. It perfectly highlights just how adaptable microbiologists have to be. The core goal, getting the cells flat and isolated, is exactly the same. But the physical technique shifts entirely depending on the organism and the specific tools you have available in the lab.
So what's the big takeaway here? Well, the next time you scoop up some soil or mix yeast into your bread dough or maybe even just touch your own desk, I want you to look at your surroundings a little differently. Through clever extraction, rigorous math, and really meticulous physical techniques, scientists can conquer the chaos of the microscale. And I think it leaves us with one incredible question to ponder. What invisible, bustling world is thriving right beneath your fingertips today, just waiting to be made visible?